tonight. We are lucky to have Nate Green, a PhD student, and he's working at WSU involved in pollen wasp research. I found out that he became interested in insects after a summer course focused on spider biology, which is very interesting. He has been hooked on entomology ever since. And tonight he's going to be presenting on fairy bees, a large genus of small bees native to North America. Please help us welcome Nate Green.
So I'm very excited to be here tonight to talk to you guys about fairy bees and Panurgines of Washington. But before I get into that, I did want to start by talking a little bit about what we're doing here at WSU. So I'm involved with the Murray and Bossert labs, where we study the systematics of aculates, particularly bees. So if you're not familiar, systematics is studying how species or groups of species are related, and then using that to answer other evolutionary questions. We also have an awesome museum here with around 2.5 million specimens in it, if I remember correctly. And we are working on databasing all the bees. I think we have around 30,000 bees, and we've databased about 23,000 of those. So we're getting close to being done. And at some point, all of that data will be on ScanBugs, so they'll be publicly accessible. And I want to give a shout out to Kylie McGuire down here in the corner, who is one of our undergraduate databasers. She's been doing a wonderful job for us, and she should be here in the chat. So like Kate said, I study Pseudomazarus. Which have been called the other bees, which I quite like. These are vespid wasps that are solitary, unlike yellow jackets or paper wasps that build these big colonies. And they're called the other bees because they provision pollen and nectar like bees. These are specialists on penstemon and Phacelia. And even though I'm based in Washington, I don't have all that many Washington records. So if you guys see these in your yards, especially in the Cascades, please let me know. I'm always looking for more records of these. And then last thing, before I start talking about fairy bees, I'm also involved in this WASP ID course, which might be of interest to some of you. We do family level identification. We teach you family level identification, a lot about morphology, how to collect. The whole thing is asynchronous, so you can just download the lectures and watch them at your leisure. If you're interested, the website's there, or feel free to reach out to me. And I'm teaching the superfamily Proctotrupoidea, which are some really weird little wasps. So I just spent 2 slides telling you that I study wasps and I'm interested in wasps. So why am I here talking to you about fairy bees tonight? I am very broadly interested in Hymenoptera, and everybody else in my lab studies bees, so they keep trying to pull me that way. But I got into fairy bees because 2 years ago, I went to Hawaii County Park, which is near Pullman, and did some collecting on some willows right by the river, and collected a couple of fairy bees, which the scientific name is Perdita. And I decided, I'm a taxonomist, so I wanted to figure out what species they were. And I figured out there were about 9 species known in Washington, and figured, oh, this shouldn't be too hard. Well, then I found the Timberlake books. So Timberlake was a taxonomist who described a ton of species, and I think most of the species of Perdita. Between 1954 and 1980, he wrote 9 books revising 600 plus species, and it's just a mess to try to go through for the few species found in Washington. So I decided I wanted to try to make a key to the species of Washington, and I'm chugging along with that slowly but surely. And Timberlake's important for the Panurgines overall, because he also revised Proto-Andrena and New World Panurginus, which I'll talk about here in a minute. So Timberlake's really important guy, but really frustrated that I had to use these giant tomes to figure out what my bees were. but I did get there eventually. And before I really dive into this, I do want to talk about anatomy a little bit, just to get everybody on the same page. So if you were here early in the bee share, Lisa was talking about the facial fovea, which are these kind of depressions on the side of the head. And those are important for differentiating Panurgines from other Andrenids. These subantennal sutures, the lines right below the antennae, are really important. And then T7, which is the very last section of the abdomen at the top. That's also important for some of the groups of Perdita. Okay, so what is a Panurgine bee? These are in the family Andrenidae, which most people are probably familiar with because of the genus Andrena, which is very common in spring bees. So Andrena and the Panurgines, which are these in Washington, are these 4 genera up here, and the Andrena are the other group. These are all united by having 2 subantennal sutures. So that's how we know that they're all closely related, along with DNA evidence. So in Andrena, you've got these big, hairy facial fovea up here, and the subantennal sutures are usually obscured because the face is so hairy. Whereas in Panurgines, the facial fovea are either absent or really reduced, and those subantennal sutures are usually pretty visible. But the best way to separate the two is looking at the wings. So this is the marginal cell. So this is on the leading edge of the wing. In Andrena, it's rounded or kind of pointed, whereas in the Panurgines, it's really distinctly truncate. So if you have a bee with 2 subantennal sutures with a really truncate marginal cell, probably a Panurgine. So here in Washington, we get 4 tribes of Panurgines, each of which has 1 genus in it. So that makes IDing to genus fairly easy. So Caliopsis has 5-ish. Perdita has 13. Panurginus has 3, and Protoandrena has 6. But those last 2, Panurginus and Protoandrena, haven't had good revisions in a long time and are probably understudied in Washington. So those numbers are really likely to change, but Caliopsis and Perdita are probably about right. So Panurgines are almost all ground nesting. I only know of 1 species that isn't, and that's the one Perdita that nests in sandstone. But everything else, I believe, is ground nesting. Almost all the species are specialists on some kind of plant. Perdita and Caliopsis tend to be found in drier areas, so they're really diverse in the west, in the deserts, whereas Protoandrena and Panurginus are more widespread, but are still pretty common, but are still more common in Western North America. And quick note on identification, these are pretty hard to ID in the field. You often need either really good pictures like Joe takes, or to look under a microscope. My field pictures are terrible. This is a picture of a Perdita I took last year. You can barely even tell it's a Perdita. So I don't try to identify these things in the field. And then the taxonomy of these groups is really unstable. So for Perdita, we know that a couple subgenera and species groups will probably be split in the future. And Protoandrena and Pseudopanurgus, there's kind of this debate about whether Pseudopanurgus is a genus or a subgenus. For this talk, I'm treating it as a subgenus. I think that's where most of the evidence points at this point, but there's some holdouts who still believe it should be its own genus. So I'm not going to go through this whole key, but I did throw together a key for the Panurgine of Washington, Panurgines of Washington. So this will take you to genus. And even though I'm not going to go through this, I will talk about a lot of these traits throughout the talk. So by the time we're done, you should be able to run this key pretty easily. And I'll get these PowerPoints to Kate or someone so we can get them to people if they're interested. Okay, so our first group is Andrenidae. These guys were first described in the genus Andrenidae in the 1850s before some other taxonomists and came along and said, those are like Andrenidae, but not quite. We're gonna call them Protoandrenidae. And there's still a little bit of confusion around the genus limits, like I said. The natural history of these are poorly known, but they are oligolectic, so they're specialists on asters, and they tend to be summer bees when more asters are blooming. So to identify these guys, they have either 2 or 3 submarginal cells. If you find a Panurginae with 3 submarginal cells, it's going to be Protoandrenidae in Washington. They have this episternal groove, which is kind of hard to see in this picture, but it's right there running up and down the side of the bee. And none of the other groups have that. And then the wing venation is a little confusing, but this first recurrent vein, the one between the first and second submarginal cell, is pretty distant from this vein down here, which isn't the case in some other groups. So the next genus is Panurginae. These tend to be spring bees. Some of them are specialists, some of them are generalists. It's kind of nebulous. There's this one species, Panurginae occidentalis, which is a specialist on L. douglasii in Northern California in like salt marshes, which sounds really cool. There's a Washington species that really commonly visits D. glandulosa. I don't think it's a specialist on it, but if you're looking to get some pictures of these or collect some, that's probably a good place to start. And then based on iNaturalist and records in the WSU collection, it seems like these are a lot more common on the west side where it's a little bit wetter. So to identify these guys, they will always have 2 submarginal cells. That episternal groove on the side of the bee isn't there. And this vein between the first submarginal cell and second is almost touching this bottom vein. So they're very close. The males really often have yellow on the clypeus, but not in the periocular areas. Maybe a little spot there, but not too much. Okay, third genus is Calliopsis. These are pretty common in the southern part of the state. It seems like Tri-Cities. They're not too uncommon there, but they can be found up by like Spokane, kind of in the center of the state. They're often found at spider flowers and mustards. Again, these always have 2 submarginal cells. And the inner subantennal suture is really short in this group. Usually the female having 3 light markings on the face, like you can see here in Edwardsi, is a pretty good way to ID them. Unfortunately, that doesn't work for the common species in Washington. This Calliopsis personata has no light markings on the face, and that seems to be the really common one. But these distinct light bands on the abdomen are a good way to separate them. The only other group that can kind of look like that is Perdita, but the wing venation is really different. And then finally on to Perdita, which I'm going to talk about the most because it's what I'm most interested in here. These are the fairy bees. I learned today that Perdita means lost in Latin. I assume because they're so small that you can lose them easily. This is one of the most speciose bee genera in North America. There's over 640 described species. 2 weeks ago, I was in Logan, Utah at the bee lab with Terry Griswold. And just glancing through their Perdita, I would bet they have another 30 or 40 undescribed species just sitting there waiting for someone to do the work. So there's a ton of species. These are most diverse in the desert Southwest, but you can find them all across the country. Bossert et al. 2021 found that these diversified really recently. So they found that around 20 million years ago, these really started to speciate. They really started to go from just a few species to a lot of species. And 20 million years ago sounds like a long time, but in terms of, animals evolving, that's really, really fast. So there could be some really cool research to do there in the future on how these are speciating. A lot of people think these are still pretty rapidly speciating. There's a lot of cool research there. These are all across Washington, but they're a little more common in central Washington and eastern Washington. They're specialists, and I'll talk more about flower associations when we get into more detail. And they tend to be summer or fall bees, but there is one group that pretty commonly occurs in the spring. So often if you find a Perdita in the spring, it's going to be Perdita in the Spherulaceae group. So I put together this little heat map of where the most species of Perdita occur. And California by far has the most. There's probably 500 species of Perdita in California. And the further you get away from there, the less species you're going to find. So Washington has about 13, Florida has 17, and I think Maine has 2. I think somewhere like Illinois has maybe 15 or 16. So to identify these guys, really you want to look at that marginal cell again. It is really short, so it's not reaching more than half of the distance from the stigma, which is right here, to the wingtip. This first submarginal cell is always longer than the second, and the easiest quick way to ID these is the thorax is usually metallic while the abdomen isn't. And off the top of my head, the only other group I can think of that's like that is Agapostemon, and those are very, very different. So like I said, there's 13 confirmed species in Washington based on my list and Karen Wright's list. And Terry Griswold tells me there's 2 or 3 more undescribed. There are 3 confirmed subgenera in Washington, and there's 1 that's a strong maybe. But our confirmed subgenera are Perdita cockerellia, which we have 2 species out of about 25. Perdita, which we have 9 species out of 400-ish. And then Perdita pygoperdita, which 2 species out of, I think, around 100. But this Perdita group is just giant, so it's split up into species groups to try to make things a little bit easier. And this is the group that's very likely to be split in the future into multiple subgenera. I put together this quick Perdita key to the subgenera of Washington. Again, not going to go through it, but I will talk about a lot of these traits. So the first group to really dive into is Cockerellia. These are large species, usually 7 or 8 millimeters, which doesn't sound very big, but for a Perdita, that's massive. The hind tibia have these thick, dense white hairs in both males and females. And in the females, like you can see here, they're kind of kinked and curled. And the wing veins are very white. The wings are really distinctly white. We only get 2 species here. The way to separate them is looking at the top of the head and, deciding if it's dull or shiny. And based off of what we have here at WSU, it seems like lingualis is way more common than albipennis. So these are often found at sunflowers and other composites from July to September. So they're really not out until sunflowers start blooming. P. lingualis is known to be communal, one of the few communal Perdita. So they share a nest entrance with a bunch of other females and then build their nests off of that. And those are reused from year to year by different generations. The albipennis is actually solitary. It doesn't ever share nest entrances as far as we know. And it takes one of every 3 days off to just kind of chill, which I like. You know, work-life balance is important. All right, next group is the giant subgenus Perdita. For these, the abdomen is usually yellow, but there are a couple of groups where it's pretty green. The scopa are much thinner and simpler. So you can see here, the hairs aren't branched, they're not curled, simple. And these visit, depending on what species you're talking about, they visit a couple different things, but rabbit brush, curly cup gumweed, and willows are really good places to find them. I think I've collected Perdita off all three now. All right, the last subgenus is Perdita pygoperdita, which is probably my favorite. With these, they have these really dark abdomens with interrupted yellow lines. Much like Perdita, the scopa is thin and simple. The clypeus, which is this part right here, has these really distinct teeth off the bottom. And then the males have that T7, the last section of the abdomen is bilobate. And I'll show you pictures of that here in a second. These are found at lilies and buckwheat, depending on the species. So Perdita nevadensis is found at buckwheats. Identifying the females is a little funky because there's 2 subspecies. But identifying the males is pretty straightforward. You're looking at these lobes on T7. So this is like a top-down view of the bee's butt. And you're deciding whether this lobe is equal to the space between it or narrower. So in Perdita wyomingensis, it's narrower. In Perdita nevadensis, it's the same. So Perdita wyomingensis is associated with lilies and buttercups. I think last summer I was out hiking with a botanist friend looking for some rare lilies in the Snake River Valley. And we found the lilies, which was super cool. And I found a bunch of Perdita wyomingensis on them. So for these, the scutum is green with a little bit of black in the middle pretty often, but it's pretty distinctly polished at the center. And then, like I said, the males, you know, you're looking at the lobes versus the space between them. Here's a side-by-side of nevadensis and wyomingensis, if anyone's going to try to ID these under a microscope. Okay. So I talked to you guys a lot about taxonomy. I wanted to kind of end here with a fun fact. The smallest bee in the world is a Perdita minima, which I really love the way this looks with its weird little orange body. Only found in California and Arizona, unfortunately not here. If anybody ever finds some, please send me some. I would love that. So thank you to the lab for their support and advice. Thanks to the M.T. James Museum and the William F. Barr Museum for specimen loans. And especially thank you to Lisa Robinson and Kylie McGuire for going out and finding me specimens specifically for me. That was great. Thank you so much.